So the focus of my laboratory has really been to develop technology for embryo selection. And we obviously all know by now that aneuploidy is very common. It's actually the most common genetic abnormality in humans. So it makes sense to focus on this as sort of the lowest hanging fruit when it comes to embryo selection to improve outcomes for patients. Obviously, we've been moving away from blastomere biopsy and fluorescence in situ hybridization to trophectoderm biopsy and comprehensive chromosome screening methods, which are what I'm going to be talking about today. So one of the methodologies that's been developed for looking at all 24 chromosomes is single nucleotide polymorphism arrays. And there is a quantitative approach that doesn't require genotyping information, where the embryonic DNA is amplified after adding an adapter, which can then be used as template for a PCR reaction. That DNA is then labeled and hybridized to an array with, in some cases, hundreds of thousands of probes across the genome. In parallel, a normal sample can be processed, labeled, and evaluated for computational comparison of the signal intensities across the genome. You can then get copy number assignments for all of the chromosomes. This is an example where you see a gain of chromosome 20 shown here. In my lab, we've done quite a bit of validation of this technology before clinical application. Some of the publications are shown here. And initially, we developed this so we could fingerprint embryos when you transfer 2 and 1 implants, and you can determine which one it was, so then you can go back and ask whether or not there were differences amongst the embryos that could have been used to select that particular one with reproductive potential. Preclinical validation included using what we refer to as a positive control. These are cell lines with known abnormalities. We think this is an important step in the preclinical validation of any assay. In this situation, you can see a variety of aneuploidies in the cell lines. This is when you use a large quantity of material, and you can see the expected gains and losses associated with those known karyotypes. And when you process single cells from the same cell line, again, you can know what to expect and make sure that the test actually gives you the expected result. Obviously, we've done some clinical trials with this methodology. It's been applied to polar bodies, translocations, and some interesting biology. But ultimately, we and others have come to the conclusion that while arrays are useful, they're relatively slow, expensive, and labor-intensive. And one of the technologies we developed was to incorporate quantitative real-time PCR because it could be performed within 4 hours in a multiplex reaction. Again, we published quite a few papers performing preclinical validation. But essentially what we're doing is amplifying different regions. This is an amplification curve where you detect the signal intensity over each cycle. That's the real-time component of the methodology. And different amplicons will amplify sooner and others later, depending on what their original quantity was in the embryo itself. You can do that on the embryo biopsy and then run the same assays with a known normal. This is just showing that you get these delta CTs, so you can compare your particular target chromosome to normalization chromosome. And in parallel, you can evaluate known normal samples and then compare these relative quantitation measures to accurately quantify the copy number of each chromosome. This is showing a heat map with copy number states from 0 to 4.8 for 4 different assays on each of the 24 chromosomes, 1 through X and Y. This is an example of a sample from a cell line that we continue to use as a quality control reagent to make sure that we actually do detect trisomy, in this case of 16 and 21, and monosomy of X and Y because it's male. And you can get a copy number plot such as what's shown here. Again, preclinical validation involved looking at positive controls using cell lines as well as rebiopsies from embryos that had been tested by SNP array originally. Consistency with cell line karyotypes is shown here. And then within embryo rebiopsies, the consistency, again, shown here. Array CGH is similar in that it uses probes to evaluate the signal intensity across the genome. In this situation, you again perform whole genome amplification where you fragment, ligate, and amplify. then label with one dye. You do the same thing with control DNA that's known to be normal, but here you label it with a different dye. You then mix the two samples, and they compete for hybridization. Wherever you see more of one sample, you get a different color, green in the situation of a gain of material, or red in the situation of loss, or yellow where your embryo is normal. And this is an example of a plot showing a gain of this particular chromosome. I think one of the things we were able to do that many groups haven't been able to is cross-evaluation of different platforms, mainly because we haven't necessarily commercialized qPCR technology, and others are commercially available and can be tested in parallel. And what we did was to rebiopsy embryos that had been given an abnormal diagnosis with the aCGH, in this case a trisomy of chromosome 22. The blinded reanalysis of qPCR was done at a collaborator's laboratory in Italy, and SNP array on a third rebiopsy if there was a discrepant result in my laboratory in the US. And you can see that in this situation, this would have been considered a false positive because both these platforms gave the same result of a normal female. Overall, when we look at a combination of two phases of that study that we published, approximately 9% false positive rate with the aCGH, which is problematic, particularly when you're thinking about evaluating the prevalence of mosaicism. If you have a 9% false positive rate, once you evaluate more than 10 samples, every embryo is going to have some type of aneuploidy. Whereas we had a 2% false positive rate with qPCR in this study, and that was significantly lower than with the aCGH. There's also been some studies on clinical outcomes, one in JARG using a aCGH showing a false negative rate of 1.5%. This is in ongoing pregnancies, the rate at which you find aneuploidy despite having transferred what you predicted to be euploid, and 0.13% with qPCR. One of the reasons we think qPCR is more accurate is that we've developed chromosome-specific cutoffs instead of just using a universal cutoff for diagnosing aneuploidy. You can see here we used actual data from embryos previously diagnosed as aneuploid by a different technology, rebiopsied them, and evaluated their copy number, which is shown here, so that now we can, instead of using a universal cutoff, develop chromosome-specific cutoffs, which may improve the accuracy of the test. And so a lot of what I've talked about are relatively obsolete technologies now. Most groups are moving to next-gen sequencing, as we've heard quite a bit about today. Most methods of chromosome screening involve the initial step of whole genome amplification, which you can then perform SNP array, aCGH, or next-gen sequencing. But also, there is the possibility of doing more targeted amplification, and then qPCR or targeted next-gen sequencing, which I'll talk about a little bit as well. And you can see from left to right that this is the trend is to try to decrease costs, and these platforms which do targeted PCR instead of whole genome amplification are relatively less expensive to perform. And there are a variety of commercial names for some of these methodologies. We've heard about carrier mapping, and Dr. Hughes mentioned a method that requires parental analysis, parental support, and a method that I talked about, which is quantitative, all of which use a SNP array platform to characterize genotypes or signal intensities for copy number in parallel. aCGH uses whole genome amplification, and the most common platform is 24-SURE. There are a variety of platforms for whole genome amplification-based next-gen sequencing, VeriSeq and ReproSeq. We've heard about VeriSeq primarily today. A research foundation now performs what's called Select CCS, which is a qPCR methodology, as well as Next CCS, which is a targeted next-gen platform. And then a group, Good Start Genetics, performs something called EmbryoView or EmbryoView with targeted next-gen sequencing. So I wanted to just review again some of the basics behind next-gen sequencing. With conventional Sanger sequencing, you have one sample and you get one sequence, and this is a combination of results from many cells and many molecules of DNA. Instead, with next-gen sequencing, we get many of these individual reads, per molecule. Those reads are then aligned to the genome. In this situation, 6 reads aligned to this position and 10 reads aligned to this position. So you count the number of reads which align to different locations in the genome, which can be represented here as gray bars. So for example, all of these reads align to chromosome 1. Then you do the same thing for the remaining 24 chromosomes. And you then normalize those reads from the embryo to counts that you've generated from reference normal samples to get a comprehensive chromosome screening diagnosis. The main reason why NextGen is less expensive is that we can barcode each individual embryo biopsy DNA PCR product and then pool them all together in order to run them on the same sequencing run in parallel. And then you can break them back out. So these are barcodes that you can attach. For example, barcode 1 to the DNA from embryo 1 and barcode 2 to embryo 2. Mix the two together and they go on the same chip. And that's really how we save in terms of cost per embryo, which is becoming a big issue as we move forward with this technology so that more patients can afford to do it. And I'll just briefly go through some of the validation that we did with a particular methodology of next-gen sequencing, which, again, we went through a phase where we evaluated cell lines, but also rebiopsies that were blinded to the original prediction. And we had relatively high concordance with the original diagnosis after unblinding the samples. Now, these preclinical validation steps are important, and you'll probably hear more about randomized clinical trials and also what we refer to as a non-selection study. We want to make sure that embryos we diagnose as abnormal don't actually possess reproductive potential. In this particular study, we looked at next-gen sequencing. We biopsied embryos and transferred based on standard criteria of selection, and then unblinded the prediction of whether or not they were aneuploid. And in all of the aneuploid embryo transfers, none of them actually sustained implantation. And we had 66% sustained implantation of embryos predicted to be euploid with this methodology. And I think this is an important study to really evaluate what the impact of mosaicism might actually be when you look at whether or not embryos predicted to be abnormal actually have reproductive potential. And as we've heard, I think a little, there is interest in looking at segmental aneuploidy associated with errors during development of the embryo. One of the important aspects to look at is whether or not the original diagnosis is predictive of the remaining embryo. This is an example where the original biopsy predicted a segmental error. We performed multiple rebiopsies and performed randomized blinded analysis. We designated the results into different categories. One is where all of the results were the same, therefore uniform segmental aneuploidy in that embryo. In some cases, we had reciprocal errors. So the original, for example, on X here, you can see a loss of material on this end of chromosome X, and the inverse or reciprocal gain of this portion of chromosome X here. So reciprocal segmental. Some cases we only observed it in the original biopsy and none of the rebiopsies. And then other cases it was confirmed in at least one of the rebiopsies. So this again has to do with the predictive value of the original test. I don't know why I'm having trouble with this. And this is just a summary of the results. You can see in 3 of the pies in this chart, the original prediction was confirmed, so we had 88% predictive value of the remaining embryo. There were 12% that were only found in the original biopsy, and these may in fact be artifacts of the method or true mosaicism that wasn't confirmed in any of the remaining embryo. But I think what's interesting is that the majority of these errors were associated with a mitotic origin in that they were not uniform throughout the embryo, which really, I think, suggests that we need methods like we've been talking about that are capable of detecting mosaicism within a trophectoderm biopsy where you might expect only a portion to possess things like segmental aneuploidy. And so I'm going to get into a little bit about mosaicism. And first just to point out that we have this sampling issue where clinically we're only going to be able to take one biopsy. we're not going to be able to evaluate the entire embryo. So there's always going to be a sampling error that's associated with trying to predict mosaicism. But in addition to actual biological variation across the embryo that's really due to mosaicism, there's technical inconsistencies we really need to consider. And this is an example where actually none of the embryos had consistent aneuploidy in all of the cells evaluated, which really goes against what we know about the origin of aneuploidy being primarily meiotic, in that we should see the same abnormality in all the cells, at least in a few of these embryos, which we didn't. And so it really questions the validity of the technology used. Another example where there was probably over-interpretation of the data suggesting self-correction. There was 0% consistency between day 3 and day 5, probably associated with errors in the original diagnosis of single cells. And we heard about the idea of using a model to make artificial mixtures. I think one of the examples was where whole genome amplified DNA from a normal and an abnormal was mixed. Here we actually mixed together cells with either a normal karyotype or trisomy 18 karyotype at different ratios. And we didn't just do one of each of those mixtures, we did multiple replicates because we really wanted to establish what the sensitivity and specificity of predicting abnormalities in a mosaic mixture actually were. And we evaluated 3 different methods recently published in JARG. One involved qPCR, and you can see that, for example, in these black bars, that as the percentage of aneuploidy increases in the mixture, the qPCR method is actually capable of detecting the abnormality. And when you get over 50%, as you might expect, more than 50% of the time we actually do detect aneuploidy in these mixtures. And the same was true with sort of what we refer to as default VeriSeq analysis, whether or not aneuploidy was predicted in these mixtures or not, just using the standard methodology of analysis with what comes off the machine when performing VeriSeq. And also recently there was a set of criteria published by another group where they interpreted the data differently by sort of resetting thresholds to call something mosaic or not. And you see, in fact, this increases the sensitivity so that you do predict aneuploidy in a significantly higher percentage of the cells at these lower levels of mosaicism or aneuploidy. But unfortunately, when you do that, you also have to evaluate the specificity. So how often do you call something aneuploid when it's really not? And you can see the overall specificity with the default VeriSeq and qPCR methods 100% of the time you don't actually have a false positive, whereas 33% of the time with these customized settings you have a false positive. And this is an important concept. You can see some examples here where you would have a false positive here, and here's a false positive, here's a false negative. So as you increase the sensitivity to predicting abnormalities in a mosaic sample, you also decrease the specificity and therefore increase the false positives that you produce, which I think, again, is important, that we have to realize some of these technologies may produce false positives, and a test that results in predicting mosaicism, the embryo may actually be completely normal to begin with, and this is potentially why we start to see outcomes such as this, where you do see successful results or outcomes after transfer of embryos that were predicted to be mosaic. I think it's a mistake to say that mosaic embryos were transferred and led to delivery of normal babies. We actually don't know if these embryos were mosaic to begin with. In fact, the technology used could have produced a false positive rather than actually having predicted accurately that there was mosaicism in these embryos to begin with. And in the validation phase of this study, they didn't evaluate the specificity or whether there were any false positives produced in the control cell line. So again, how often did their criteria for calling something mosaic inaccurately predict mosaicism of a chromosome that was known to be normal to begin with? And I also wanted to bring up this point with respect to how we are classifying embryos as mosaic. When you are able to dissociate the entire embryo, it's a little easier to develop more rigorous criteria. For example, seeing reciprocal aneuploidy here at monosomy of 15 and trisomy of 15 in the same embryo. This is probably the strongest evidence for actually having mosaicism in the embryo. Or at the single cell level, you can also have confirmation in 2 different biopsies of the same abnormality. All the way down to what we see here, which is what's being used clinically, and this is sort of intermediate ratios that fall in a so-called mosaic range. This is the evidence used to report mosaicism in current practice, and it's actually the weakest evidence for whether or not there's mosaicism actually present in the embryo. There are other explanations in these situations where you see these intermediate log2 ratios or copy number assignments that don't actually fall in what you might consider a normal range or the uniform aneuploid range. And when you only have copy number analysis, you might interpret this as a mosaic embryo. But actually, these are samples where we purposefully contaminated them to show that they can contribute to these intermediate levels. And without having genotype data, which you actually can produce with an alternative methodology, you would probably predict this embryo to be mosaic. But with the addition of genotype information, what you normally would see is a 50-50 ratio of these single nucleotide polymorphisms where you have heterozygosity. You would expect the same amount of one allele as the other, so this 50% ratio. But when you start to see contamination, you can see that the data becomes blurred, and you don't really have this clustering around 50%. And this is one way to distinguish between true mosaicism and actual contamination. Another contributor to what might be predicted to be mosaic is in this situation where you do in fact have triploidy. So all of these chromosomes assigned 2 copies are actually 3. And this particular chromosome here, it's not really mosaic, it's actually 2 copies. And you only know that by looking at the allele ratio. And in this situation, you can see you have a 1-to-2 ratio of some SNPs and a 2-to-1 ratio of others. 2-to-1 allele ratios give you this spread away from 50-50 and towards 1/3 and 2/3. And so this might be interpreted as mosaic for chromosome 2, but actually it's a triploid embryo. So to conclude, I think it is important to validate these methods rigorously, and valid methods of CCS actually can improve outcomes. Targeted next-gen methods are probably the most cost-effective methodologies moving forward. If you have inaccurate methods of chromosome screening, you can overestimate mosaicism, self-correction, and actual aneuploidy. And as we develop new tools to better predict mosaicism and segmental aneuploidy, I think it's important to carefully consider not only the sensitivity, but also specificity and predictive value. Thank my collaborators and everyone at RMA, and also I don't get a chance to do this too often. My mom is here, and I want to thank her for coming with me and being such an amazing person. Thank you.